So in this video, we're going to be discussing DNA methylation and histone acetylation, and these are subtopics in a wider topic called epigenetics. Okay, so what is epigenetics? So the process of increasing or decreasing the expression of certain genes is called epigenetics. So gene expression basically means that certain genes are available for RNA polymerase to form proteins. So all cells in our body, whether skin cells, liver cells, or muscle cells, contain the same genetic material. So what makes skin cells functionally and structurally different from liver cells, and what makes liver cells different from muscle cells? And this is the process. This process is called epigenetics. So skin cells express genes that help them become skin cells. Liver cells express genes that help them become liver cells, and muscle cells express genes that help them become muscle cells, and they suppress any other genes. So muscle cells will suppress the genes that are required for liver function, and liver cells will suppress the genes that are responsible for skin cell function. Okay, so there are two important epigenetic processes that we have to learn. The first is called DNA methylation, and the second is called histone acetylation. Okay, so let's start with DNA methylation. So DNA methylation is the process of adding a methyl group onto nucleotides in DNA. Now this occurs in the segments of DNA that have repeated CG segments. So when DNA is methylated, so those genes become inactive. So the methylation of DNA inactivates the transcription of those genes. So for example, if we go to a liver cell and we look at its DNA, we're going to find that the genes that are responsible for skin cell function are going to be methylated. So they're going to be inactive in the liver cell. So the second epigenetic process that we're going to talk about is called histone acetylation and deacetylation. Now this is the process of adding or removing acetyl groups on the histone proteins. Okay, so let's start with histone acetylation. So right here we have something called closed chromatin. So this is basically chromatin that is condensed together. So basically RNA polymerase can't really come along and read the DNA here because it's so closely condensed. So what happens here is that we get an enzyme called HAD, and HAD is going to come along and it's going to add acetyl groups onto the lysine amino acid of the histone proteins. And this is going to lead the chromatin to become more relaxed, so DNA polymerase can actually come along and start transcribing the genetic information. So histone deacetylation is the reverse process in which we remove the acetyl groups from the histones, and that's gonna turn the chromatin from a relaxed form into a more condensed form. So deacetylation actually prevents transcription of genes, while acetylation increases the transcription of genes.